The patient is in the room. They have a fever, maybe a rash, some confusion. You suspect an infection. You do what you've been trained to do. You draw blood, you send it to the lab, and you wait for the culture. But here's the problem. The culture comes back negative. Nothing grew.
The silent lab report. It's the nightmare scenario for a third-year medical student on the wards. You know something is wrong, but the agar plate is just empty.
Exactly. Because maybe the organism isn't there anymore, or maybe it's Treponema pallidum, the agent of syphilis, and it simply refuses to grow on your standard blood agar. It's notoriously difficult.
Well, it's a picky bug,
Right.
or maybe it's a virus that needs living cells to replicate. You can't just put it on a plate of jelly. You can't hunt the beast because, for all intents and purposes, the beast is invisible.
So you stop hunting the beast.
And you start tracking the footprints. That is the absolute core of what we're doing in this deep dive. We are tearing apart Chapter 6 of Murray's medical microbiology and layering in some really important clinical pearls from Chapter 47 of Jowet's. We are talking about serologic diagnosis.
And this requires a fundamental mental shift. I mean, up until now, in bacteriology, we've been obsessed with the organism itself. Gram stains, shapes, metabolism, toxins, all of that. Today, we don't really care about the bug directly. We care about the host's reaction to the bug. We are, as Murray puts it, analyzing the black box.
I love that metaphor. The patient's serum is a black box. You spin down the blood, you get this clear, straw-colored fluid, and to the naked eye, it's just liquid. But immunologically, it is a historical archive.
Yeah.
It contains the record of every pathogen that patient has ever fought and a real-time status report of what they are fighting right now. It's all there in the antibodies.
So our mission today is to build a mental model of how we peer into that black box without, actually opening it. We need to understand the tools, the physics of how we visualize these invisible reactions, and most importantly, the logic of interpretation.
And that interpretation piece is so, so key. Because serology is indirect evidence. It's like convicting a suspect based on their credit card bill rather than catching them at the scene of the crime.
That's a great way to put it.
You have to be careful. You have to build a case. You can't just rely on one piece of data.
So let's start with the tools, the probes. If we want to find a specific needle in the haystack of proteins in the blood, we need a magnet.
And that magnet is the antibody. But as Murray points out right in the very first section of Chapter 6, not all magnets are created equal. You have your shotgun and you have your sniper.
Polyclonal versus monoclonal antibodies.
Okay.
Let's start with the shotgun. This is the polyclonal antibody. This is historically how the whole field began, right?
Right. Poly, meaning many. Imagine you want to create a test for, say, group A strip. You take an antigen, maybe a protein from the bacterial capsule, and you inject it into an animal, like a rabbit.
Okay.
The rabbit's immune system recognizes that protein as foreign and launches a defense. It activates B cells.
But it doesn't just activate one B cell. That's the key, isn't it?
And that's the absolute key. That bacterial protein is a large, complex, three-dimensional structure. It has corners, bumps, ridges, a whole landscape. We call these specific shapes epitopes.
Right, the antigenic determinants.
Exactly. And the rabbit's immune system attacks all of them simultaneously. One B-cell clone makes an antibody against the top corner. Another B-cell clone makes an antibody against the bottom ridge. A third targets a little loop on the side.
So when you harvest the rabbit serum a few weeks later, you have a cocktail. You've got, thousands of different antibody populations, all targeting the same antigen but grabbing it in different spots.
Since the shotgun, it just sprays the target.
And what's the clinical upside of a shotgun? Why would we want that?
Sensitivity. It is incredibly hard for the bacteria to hide from a polyclonal mixture. Think about it. If the bacteria mutates one little corner of its protein, the other 999 antibodies in the mix will still latch on. You won't miss the diagnosis just because of a minor antigenic variation. It's a very robust way to detect something.
But there's always a trade-off in medicine. So what's the price you pay for that sensitivity?
Specificity. Because you have such a diverse crowd of antibodies, there's a higher statistical chance that one of them might accidentally latch onto something else.
Something is not supposed to.
Right. Maybe a protein from the patient's own cells or a different harmless bacteria that just happens to look somewhat similar at one epitope. That's cross-reactivity. That's a false positive.
Okay, so that's the shotgun. Great for finding the target, but maybe a little messy. So if we need precision, if we need to know exactly what we're looking at with zero noise, we need the sniper, the monoclonal antibody. And this is where Murray gets into the weeds of hybridoma technology.
And this is seriously high yield for the boards. It's not enough to just know that monoclonal antibodies exist. You really need to understand the cellular engineering, the factory that builds them. It's a beautiful piece of science.
So walk us through the logic here. The goal is one single antibody that targets one single epitope. That's it.
Correct. So you start by immunizing a mouse. You inject it with your antigen of interest. Then you take the spleen cells, which contain the B cells. You isolate the specific B cell that makes the exact antibody you want. You found your master craftsman.
But B cells are mortal, right? If you put a normal B cell in a petri dish, it dies in a few days. You can't build a global diagnostic industry on a cell that dies over the weekend.
No, you can't. It's not sustainable. So you have to make a deal with the devil. You fuse that precious B cell with a cancer cell, specifically a myeloma cell.
A plasma cell cancer.
Right. Myelomas are immortal. They are programmed to divide forever. But the myeloma cells we use for this process have two very specific engineered traits. First, they can't make their own antibodies. They're like factories with all the machinery running, but no blueprints.
So they don't contaminate the final product.
Exactly. And second, and this is the really clever part, they are missing a key enzyme for DNA synthesis.
This is the biochemical trap.
It is. The enzyme is HGPRT, hypoxanthine guanine phosphorybosyltransferase. It's part of what's called the salivage pathway for making nucleotides. Normal B cells have this enzyme. The special myeloma cells we use for fusion do not.
So you have these two cell types. You mix them together in a tube. You use something like polyethylene glycol to sort of melt their membranes together, and you end up with a soup containing three things: unfused B cells, unfused myeloma cells, and the fused hybridomas.
Now comes the selection pressure. You have to kill off the cells you don't want. So you put this soup into a special broth called HAT medium, hypoxanthine, aminopterin, thymidine.
And this is where the carnage happens.
It is. The aminopterin in the medium blocks the main pathway for DNA synthesis, the de novo pathway. So to survive, the cells must use the salvage pathway.
Okay, so let's track the fates of the three cell types. What happens to the unfused myeloma cells?
They lack the enzyme HGPRT, so they can't use the salvage pathway. The main pathway is blocked. The backup pathway is broken. They starve for nucleotides. They die.
Gone. Okay, what about the unfused B cells?
They have the HGPRT enzyme, so they could survive the medium, but they are mortal. They just die of old age after a few divisions. They can't keep going.
So the only thing that survives is the hybridoma.
Exactly. It's a perfect chimera. It gets the HGPRT enzyme from the B cell, so it survives the HAT medium. And it gets the immortality from the cancer cell, so it lives forever. It's the only cell that has both things it needs to thrive in that specific environment.
That is natural selection in a petri dish. It's brilliant. And the result is a colony of cells that pumps out one single, identical, pure antibody, the sniper shot.
Infinite supply, perfect specificity. This technology absolutely revolutionized medicine. And not just for diagnostic testing, but for therapeutics.
Right, the MABs.
Anytime you see a drug ending in mab, rituximab, trastuzumab, adalimumab, that is a monoclonal antibody made by a hybridoma or a similar technology. It's a sniper rifle designed to target a cancer cell or an inflammatory molecule.
Okay, so we have our tools. We have the shotgun, which is polyclonal for sensitivity, and we have the sniper, which is monoclonal for specificity. Now, how do we use them to see the invisible? Murray organizes this by the nature of the reaction. And the first one is all about physics. Precipitation.
This brings us to what I like to call the dinner party problem, or more formally, the concept of lattice formation.
Explain that, because intuitively you'd think more antibody plus more antigen equals more reaction. But that's not true here.
It's not linear at all. To make a visible reaction, a precipitate that literally falls out of solution, you need to build a giant 3D structure. You need a lattice.
A web?
A web, exactly. Imagine antibodies are Y-shaped. They have two hands, right, as the IgG. And antigens usually have multiple places to be grabbed, multiple epitopes, so you need them to hold hands in a giant chain. But this depends entirely on the ratio. This is the zone of equivalence. If you have way too much antigen, let's say 100 antigens for every one antibody, the antibody grabs two antigens and its hands are full, it's saturated, it can't grab anything else to link the chain to another antibody. You get tiny soluble complexes, no web, no precipitate.
Okay, that's antigen excess.
Okay.
Prozone of antigen excess.
Now flip it. You have 100 antibodies for every one antigen. The antigen gets swarmed. It's covered in antibodies like a dog pile. But again, the antibodies can't reach out and grab another antigen because there aren't enough to go around. they're all stuck on the same guy.
Still no web.
No web, no precipitate. This is antibody excess, and this is the one that's clinically dangerous. This is the prozone phenomenon.
And this is a classic board question, isn't it?
An absolute classic. Imagine a patient has secondary syphilis. Their bacterial load is high, and their immune response is massive. Their antibody levels are sky high. You run a screening test like VDRL or RPR, which relies on flocculation, a type of precipitation.
So you add the test antigen to their serum.
And because there's so much antibody, you get the prozone effect. The antibodies swarm the antigen, but don't form a lattice. The test looks negative.
A false negative because the patient is too positive.
Exactly. It's this bizarre counterintuitive result. The lab tech looks at the clear tube and thinks patient is healthy, but actually they are highly infectious and need treatment urgently.
So how do you fix it?
You have to dilute the patient's serum. You literally water it down 1 to 10, 1 to 100. You dilute it until you hit that sweet spot, the zone of equivalence. That's where the ratio is just right, maybe 1 to 1. The antibody grabs an antigen, which is grabbed by another antibody, and the lattice grows and grows until, boom, it becomes visible.
And this lattice formation, this principle, is the basis of the Outreloni test, which frankly looks like a geometry puzzle from the 1970s when you see a picture of it.
It is old school, but the boards love images of it because it tests your understanding of homology and identity in a very visual way. You have a gel plate, like agar. You punch holes in it. You put antibody in a central well and maybe 2 different antigens in wells above it.
And they just diffuse toward each other through the jelly.
Right. They creep through the agar. Where they meet at the zone of equivalence, you get a white line, a precipitin line.
And the shape of that line tells you everything. You have 3 patterns you have to know: identity, non-identity, and partial identity. Let's visualize this for the listener.
Okay, pattern 1. Identity. You have 2 antigen wells side by side at the top and 1 antibody well at the bottom. If the 2 top wells contain the exact same antigen, the antibody recognizes them equally. The 2 diffusion fronts will meet the antibody front and merge perfectly. It looks like a smooth, continuous arc or a bridge connecting the 2 antigen wells.
A smooth arc equals identity. It's the same stuff.
Yep. Pattern 2, non-identity. Now, the 2 antigens are totally different. Let's say one is from fungus A and the other from fungus B. The antibody mixture might recognize both if it's polyclonal, but they are separate events. The precipitin lines don't merge. They completely ignore each other and cross over. It looks like an X or cross swords.
An X marks the spot for different. Non-identity. Easy enough.
Pattern 3, partial identity. This is the tricky one that trips people up. Here, the antigens are mostly the same, but one is a bit more complex or has an extra epitope.
Okay.
The lines will merge because they share common epitopes, but there's a little tail, a spur, that sticks out past the junction, and that spur always points toward the simpler antigen. It basically means the antibody found something unique in the more complex sample that the simpler one lacks, like an extra reaction happening.
So you have to look for the spur and which way it's pointing. Is this actually used in real life anymore? Or is this just for the exam?
It's mostly historical and for teaching, except for mycology. Jawitz specifically calls this out for fungal infections. We're talking about things like histoplasma, blastomyces, coccidioids. If you see an exam question with a picture of an immunodiffusion plate or it mentions precipitin bands in a patient with a chronic lung infection, you should absolutely be thinking about a systemic fungus.
Good to know.
Yeah.
Okay, another precipitation-based technique is radial immunodiffusion. How is that different from Outreloni?
It's simpler and it's quantitative. In this case, the antibody isn't in a well. It's mixed into the entire agar gel from the start.
So the whole plate is full of antibody.
Right. Then you just punch one well in the center and you add your antigen, say, the patient sample. The antigen diffuses outwards in a circle, and as it spreads, it forms a precipitin ring where it hits the zone of equivalence with the antibody in the gel.
So you get a circle like a target.
Exactly. And the key metric here is that the diameter of that ring is proportional to the antigen concentration. A bigger ring means more antigen was in the well to begin with. You can measure the ring and compare it to standards to get an exact concentration. It's used to quantify things like immunoglobulin levels, IgG, IgA, IgM, in a patient's serum.
Okay, let's move from lines and circles and jelly to something faster, something we use every single day in the clinic: agglutination.
Right. This is similar to precipitation, but instead of seeing soluble proteins fall out of solution, we are clumping particles. Things that are already big, we're talking whole bacteria, red blood cells, or most commonly, latex beads.
This is the basis of the rapid strep test, right? The one they do in the pediatrician's office.
Exactly. That is latex agglutination. You take tiny microscopic latex beads and you coat them with specific antibodies against group A strep. You take the throat swab from the kid, you extract the antigen, and you mix it with the beads on a little card.
And if the strep antigen is there?
The antibodies on the beads grab it, and since each antibody has two hands and each antigen can be grabbed by multiple beads, they pull all the beads together into a big clump.
And because the beads are white and the card is usually black, you can see these clumps form with your naked eye almost instantly.
It's instant gratification diagnostics. But there is a really interesting nuance here when we talk about hemagglutination using red blood cells as the particles. This is crucial for things like blood typing and the Coombs test. And there's a physics problem we need to solve called the zeta potential.
The force field.
Yeah, that's a good way to think of it. Red blood cells have a net negative surface charge from sialic acid residues. So in solution, they naturally repel each other, like two negative ends of a magnet. They stay apart. This repulsive force is the zeta potential.
So to get them to agglutinate, to clump together, you need an antibody that is physically big enough to bridge that gap and overcome the repulsion.
Enter IgM.
The big one.
IgM is a pentamer. It's a massive molecule, looks like a snowflake. It has a huge wingspan, 10 binding sites. It can easily reach across that zeta potential gap and pull two red blood cells together. That's why IgM antibodies, like the ones against A and B blood antigens, are potent agglutinins. If you mix anti-IgM with type A red cells, you get immediate strong clumping.
But IgG is different.
IgG is a monomer. It's tiny. It's got two hands, but its arms are short. It's often too small to bridge the gap. It might coat the red blood cells, but it can't pull them together against that repulsive force.
So you could have a situation where a patient's red cells are covered in IgG antibodies, like in autoimmune hemolytic anemia. But when you look at the tube, everything looks normal. No clumps.
A hidden positive. And a very dangerous one.
Yeah.
To see that reaction, you need a helper. You need the Coombs reagent.
What is that?
The Coombs reagent is an antibody against human IgG. It's usually made in a rabbit. You add this reagent to the patient's IgG-coated red cells. The rabbit anti-human IgG then binds to the IgG that's stuck on the red cells and links them up. It artificially bridges the gap.
So the direct Coombs test is looking for antibodies already stuck to the RBCs in the patient's body.
Precisely. It overcomes the zeta potential to visualize an invisible coating. It's an absolutely critical test in transfusion medicine and hematology.
Okay, let's level up. We want to see things that don't clump. We want to find a needle in a haystack and light it up with a neon sign. Let's talk about immunofluorescence.
Now we are swapping the lattice for a flashlight. The concept is simple. We take an antibody and we covalently attach a fluorescent molecule to it, usually fluorescein, which glows this brilliant apple green under a UV microscope.
And Murray describes two main ways to do this, direct and indirect methods. What's the strategic difference between them?
Direct immunofluorescence, DIF, is for looking for antigen in tissue. You have a physical chunk of a biopsy, say, a kidney biopsy where you suspect Goodpasture syndrome, or a skin biopsy for herpes zoster, or even brain tissue looking for rabies.
Okay.
You take an antibody against your target, an antibody that has the flashlight already attached to it, and you just dump it on the tissue slice. You wash away the excess and you look under the microscope. If it glows, the antigen is there.
Simple. One step. Direct.
But it's not very sensitive. You only have one flashlight per antigen. If the infection is low level or there's not much antigen there, you might miss it. The signal is weak. That's why for serum testing or anytime we need more sensitivity, we prefer indirect immunofluorescence.
Sandwich approach again. Kinda.
It's an amplification strategy. Here, the first antibody, the primary antibody that finds the target, has no flashlight. It's just a normal antibody from the patient's serum, for example.
Right.
Then you add a secondary antibody, an anti-antibody, that carries the flashlight. For example, a goat anti-human IgG antibody with a fluorescent tag. This secondary antibody then binds to the primary antibody that's already stuck to the target.
And the magic is amplification.
That's the whole point. Because the primary antibody is a large protein molecule, multiple secondary antibodies can grab onto it. So for every one virus particle, you might now have 5 or 6 flashlights pointing at it instead of just one. It makes the signal much, much brighter and easier to detect. It's a more sensitive test.
Now let's take that concept, fluorescent tags on antibodies, and put it on steroids. Speed it up and put it on a conveyor belt moving at 5,000 cells per second. That's flow cytometry.
Flow is the absolute workhorse of modern immunology and hematology. It's an incredible technology. You take cells in a liquid suspension, like blood. You force them through a tiny nozzle, so they have to line up single file. And as they fly through, one by one, you shoot a laser beam at them.
And as each cell flies through that laser, the machine measures a few things. This is physics again.
It is. First, it measures the shadow the cell casts. How much light did the cell block? The detector is right in line with the laser right behind the cell. This is forward scatter, or FSC. The bigger the shadow, the bigger the cell. Simple as that.
So forward scatter equals size. Got it.
Again, it measures the light that bounces off to the side, the refraction and reflection from the internal components of the cell. This is side scatter, or SSC. This tells you about the internal complexity. Is the cell smooth and simple like a lymphocyte, which is basically a bag of nucleus? Or is it full of granules and a complex nucleus like a neutrophil?
So side scatter is granularity. That means just with those two physical measurements, size and granularity, you can already start to tell the different blood cells apart.
Exactly. You can make a plot of forward versus side scatter. A lymphocyte is small and smooth, so it has low forward and low side scatter. A neutrophil is bigger and very granular, so it's high forward and high side scatter. You can already sort the major populations just by physics.
But then comes the third measurement, the really powerful part, fluorescence.
This is where the specificity for monoclonal antibodies comes in. If you've pre-tagged those cells with specific monoclonal antibodies, say an anti-CD4 antibody with a green fluorescent tag and an anti-CD8 antibody with a red tag, the laser will excite those tags as the cell flies through.
And the machine counts them. Green, red, both, neither.
It generates a dot plot. This is the classic image you'll see on the USMLE or Comlex. Each dot on the screen represents a single cell. You can use the computer to draw a box around the cluster of lymphocytes. That's called gating. And you can ask the computer, okay, inside this box of lymphocytes, how many are green? How many are red?
And that gives you the precise CD4 count for an HIV patient. It's quantitative, it's rapid, and thanks to those monoclonal antibodies, it's incredibly specific.
It's the gold standard for immunophenotyping, for leukemia and lymphoma diagnosis, for all sorts of things. It's a cornerstone of the modern lab.
Okay, we have to talk about the test that everyone knows by name, even if they don't really know how it works. ELISA.
Enzyme-linked immunosorbent assay. It's a mouthful, but the logic is actually quite elegant. It's just like immunofluorescence, but instead of a flashlight, we're using an enzyme as our reporter.
Why swap the flashlight for an enzyme? What's the advantage?
The advantage is signal amplification, again, but in a different way. And also, it's easier to read. Enzymes are catalytic multipliers. One single enzyme molecule, like horseradish peroxidase, can chew up thousands and thousands of substrate molecules in a few minutes.
And what does that do?
It turns a clear liquid into a colored liquid, usually yellow or blue, and the intensity of that color can be precisely measured by a machine called a spectrophotometer. The darker the color, the more positive the result. It gives you a number, an optical density.
So it's not just yes, no, like looking for a glow. It's how much. It's quantitative.
Exactly. The most common version you need to know for your exams is the sandwich ELISA, which is also called an antigen capture assay. This is how we find things like HIV P24 antigen early in an infection or rotavirus in a stool sample.
Okay, walk us through the sandwich construction. What are the layers?
You start with a little plastic well from a 96-well plate. It's coated with a capture antibody. That's the bottom bun of your sandwich. It's stuck to the plastic.
Right.
You add the patient's sample serum, stool, whatever. If the virus or antigen is there, it gets stuck to the bottom bun.
Then you wash.
A crucial step. You have to wash away everything that didn't stick. All the patient's other proteins, other bacteria, everything. Then you add the detection antibody. This is the top bun. This antibody also recognizes the virus, but usually at a different epitope. And this one has the enzyme attached to it.
So the virus is now trapped or sandwiched between two different antibodies.
Exactly. You wash again to get rid of any unbound detection antibody. Then, the final step, you add the colorless substrate. The enzyme activates, the color changes, and the spectrophotometer reads the result. More color means more antigen was captured.
How sensitive is this test?
Very, very sensitive. But ELISA can be the eager beaver of tests. It really wants to be positive. Sometimes if a patient has sticky serum, high lipids, or weird autoantibodies like rheumatoid factor, you can get non-specific binding. You get a false positive. The well lights up even when the specific virus isn't there.
Which is precisely why we never diagnose HIV based on a single ELISA test. We need a backup. We need a confirmation. We need the Western blot.
Or, as Murray correctly calls it, the immunoblot. This is the confirmation. If ELISA is a mixed soup that tells you something tastes like chicken, the Western blot is the ingredients list that tells you it contains chicken breast, chicken thigh, and chicken stock.
It separates the proteins. It tells you what the antibody is reacting to.
That's the key. It take the virus, you mash it up, and you run its proteins through an electric gel. This technique is called SDS page. The proteins separate by their molecular weight. The heavy ones, like GP120, stay near the top, and the light ones, like P24, run down to the bottom. You get a ladder of proteins.
Then you have to get that ladder out of the flimsy gel and onto something solid.
Right. You transfer or blot that protein ladder onto a piece of nitrocellulose paper. It's like making a protein photocopy.
And then you dip that paper with its invisible protein bands into the patient's serum.
And if the patient has antibodies, they will stick to the specific rungs of that ladder. You'll then add a secondary enzyme-linked antibody to visualize where the patient's antibodies have bound. You might see a dark band appear at the 120 kilodalton mark for GP120, and another one at the 24 kilodalton mark for P24.
And to call it a positive HIV test, you need to see a specific pattern of bands. You need to see the fingerprint of an immune response to HIV, not just some random reaction.
Exactly, and ELISA just tells you something reacted. The Western blot tells you, specifically, antibodies against GP120, GP41, and P24 reacted. It rules out almost all of the false positives from the screening ELISA.
And for the exams, remember the mnemonic? S-no drop.
A lifesaver. S goes with D. Southern blot detects DNA, N goes with R. Northern blot detects RNA. And W goes with P. Western blot detects protein. Do not miss that free point on the exam.
All right, we have to tackle the beast. The one section in Murray that I swear makes medical students cry. Complement fixation.
Ah, yes, the resource depletion model. It's so counterintuitive because a negative result looks like an explosion and a positive result looks like nothing happened. You absolutely have to wire your brain backwards for this test.
Let's slow this down and walk through it step by step.
Yep.
Let's say we have a patient. We want to know, do they have antibodies against the influenza virus?
Okay, step 1. You take the patient's serum and you heat it to 56 degrees Celsius. You have to kill their own complement proteins. You need a controlled experiment, so you can't have their own variable levels of complement interfering with the test.
Step 2. In a test tube, you mix their heat-inactivated serum, the influenza antigen, and a very precise, measured amount of guinea pig complement. This is your fuel tank.
Now you wait. You incubate it. This is where the first reaction happens. If the patient has antibodies against influenza, scenario A, a positive patient, those antibodies will grab the antigen. They form an antigen-antibody complex.
We know from basic immunology that antigen-antibody complexes activate the classical complement pathway. They fix the complement.
They consume it, the fuel tank gets emptied, the complement is all used up in this invisible battle happening inside the test tube.
But you can't see that happen. It's all colorless.
Yeah.
You need an indicator system.
Right. So for step 3, you add your indicator. This is sheep red blood cells that are pre-coated in antibody. We call them sensitized RBCs. These are essentially little biological mines waiting to be detonated, but they need a spark. They need complement to explode via hemolysis.
But in scenario A, the positive patient.
The complement is gone. The patient's antibody-antigen complex used it all up in the first step. So the sheep RBCs sit there, they don't explode, they slowly sink to the bottom of the tube under gravity and form a tight red button.
So no explosion, no hemolysis. And that means the patient did have the antibody we were looking for.
No hemolysis, yeast positive result. It's burned into my brain.
Now let's flip it. Scenario B, a negative patient. They have no antibodies against influenza.
You do the same thing. You mix their serum plus the influenza antigen plus the guinea pig complement. But with no antibody, no antigens, antibody complex is formed. There's no battle. The complement just floats around, bored. It is not fixed. The fuel tank is still full.
Now you add the indicator, the sensitized sheep RBCs.
The free-floating complement sees them and says, finally, a target. It attacks the sensitized RBCs and activates the membrane attack complex. Boom! Lysis. The red blood cells explode, releasing their hemoglobin, and the liquid in the tube turns a clear pink or red.
Hemolysis equals negative result.
It's a double negative logic puzzle. Did the fuel, the complement, survive the first step? If yes, the patient is negative. If no, the patient is positive.
That is exactly the kind of multi-step, mechanism-based reasoning question that distinguishes a 240 from a 260 on the STEP exam. It forces you to think about the mechanism, not just memorize a fact.
Absolutely. It's a beautiful, if convoluted, assay.
Okay, we're in the home stretch now. We've built the tools. We've visualized the reactions. Now we have to talk about how we interpret the numbers. This is clinical serology.
This is where the real detective work happens. Because just finding an antibody doesn't tell you when the crime happened. Was it yesterday? Or was it 10 years ago? The presence of an antibody just tells you there was an exposure at some point.
So we talk about two main things to figure out the timing: titers and isotypes. Let's start with titer.
Titer is just a measure of concentration, but we express it in a slightly odd way. It's the inverse of the greatest dilution that still gives a positive result. We do serial dilutions of the patient's serum 1.2, 1.4, 1.8, 1.16, and so on. We run the test on each dilution. The last tube that's still positive is your titer.
So a titer of 64 means you have more antibody than a patient with a titer of 4.
Right, 8 times more. But here is the golden rule of serology that you absolutely must know. One single titer is often meaningless. If I tell you your herpes simplex virus titer is 1.32, are you sick right now? Or did you get a cold sore 5 years ago? I have no idea. The snapshot doesn't tell the story.
You need context. You need the timeline. You need more than one data point.
You need paired thera. This is the classic method. You draw blood now, during the illness, that's the acute phase sample, and you draw blood again in 2 to 3 weeks, that's the convalescent phase sample. You are looking for a change. You're looking for a rise.
And not just any rise. There's a specific number to look for.
A 4-fold rise, this is critical. A 2-fold rise, say from 1.4 to 1.8, that's just noise. Maybe the pipette was slightly off, maybe the tech had too much coffee. That's within one dilution of experimental error. We ignore it.
But if it goes from 1.4 to 1.16...
that is 2 tube jumps. That is a 4-fold increase. That is biologic reality. That proves the immune system is actively ramping up production right now. That is diagnostic of an active recent infection.
But in the ER, or even in the clinic, we often can't wait 2 weeks for a convalescent titer. We need to know now. So we look at the isotype. We look at IgM versus Ig.
Yes, the crime scene dating. IgM is the first responder. It's a large pentameric molecule. It shows up early in an infection, maybe day 5-7, and it fades away after a few months. So if you see specific IgM, it means the house is currently on fire. It points to an acute primary infection.
And IgG.
IgG is the memory. It's the archive. It takes longer to develop, a few weeks, but it lasts for years, sometimes for life. If you see high IgG and no IgM, it means old news. It's evidence of a past exposure or vaccination.
And this brings up the idea of a primary versus a secondary or anamnestic response.
Right. The first time you see a pathogen, your primary response is slow. You get a little bump of IgM first, followed by a slower, lower peak of IgG. But the second time you see that same pathogen, maybe years later, your memory cells kick in.
The anamnestic response.
And it's a completely different picture. You get an immediate, rapid, and massive spike in IgG. It's much faster and much higher than the primary response. IgM might show up a little, but the IgG response is dominant. The immune system remembers.
But there is a massive exception to this IgMig rule that Murray highlights, and it's a crucial pediatric concept: the newborn.
This is something you absolutely cannot forget. IgG is the only antibody class that actively crosses the placenta. Nature designed it this way so mom can pass her protective immunity to the baby for the first few months of life.
So a newborn baby with a high IgG titer against measles.
It's just a baby with a vaccinated mom. That is maternal antibody. It protects the baby, but it absolutely does not mean the baby is infected. That maternal IgG will fade after 6 to 12 months as the baby's body clears the mom's proteins.
But IgM is different.
IgM is too fat. It's a big pentamer. It cannot cross the placenta. So if you find specific IgM in a newborn's blood, say against rubella, CMV, or toxoplasmosis, one of the tortoise infections that did not come from mom, the baby made it.
Which means the baby was infected in utero.
Exactly. IgM in a newborn is diagnostic for congenital infection. That is a critical distinction that saves lives and directs immediate therapy.
We have covered a massive amount of ground today. I feel like we've gone through an entire immunology lab. We went from the shotgun of polyclonal antibodies to the sniper precision of monoclonals. We watched lattices form in the zone of equivalence.
We lit up antigens with immunofluorescence and sorted cells with flow cytometry. We used enzymes in ELISA and separated proteins in Western blot. It's quite the toolkit.
And finally, we solved the complement fixation puzzle and learned how to read the timeline written in the language of IgM and IgG.
It's a lot of acronyms, it's a lot of techniques, but if you hold on to that black box metaphor that we are reading the footprints of the immune system to figure out what happened, it all fits together logically.
Before we wrap up with our quiz, I want to echo a sentiment from the end of the chapter. Murray talks about diagnostic humility.
Yes, this is so important. Remember that serology is indirect. Sometimes the body is silent-like in a state called anergy, where an advanced AIDS patient is so immune-suppressed they can't make antibodies. Their serology tests will be negative, but the patient is dying of the infection.
Or sometimes the body lies.
Cross-reactivity, exactly.
Yeah.
An antibody against Epstein-Barr virus might accidentally react with something on a sheet red blood cell, or an antibody against one type of rickettsia might cross-react with another. Serology is a powerful tool, but it must always be interpreted in the context of the patient sitting in front of you. Treat the patient, not the titer.
Powerful words to live by. Okay, wipe the mental whiteboard clean. It's time to calibrate your mental titer with a rapid recall.
Let's test your retention on these key concepts.
I'm going to ask a question based on today's deep dive. I'll pause for a few seconds to let you think about the answer, and then our expert will break it down. Ready?
Let's do it.
Question 1. A patient is being evaluated for secondary syphilis and has a very high bacterial load. A VDRL test, which relies on flocculation, is performed on their undiluted serum, but no flocculation is observed. However, when the serum is diluted 1.10 and retested, the test becomes strongly positive. What is the name of this phenomenon? Pause.
That is the classic prozone phenomenon. It occurs due to antibody excess. There are so many antibody molecules that they saturate all the binding sites on the antigen, preventing the formation of the large lattice network required for visible precipitation. Diluting the serum brings the reactants back into the zone of equivalence.
Excellent. Question 2. You are creating a hybridoma to produce monoclonal antibodies. You fuse B cells with special myeloma cells and plate them on HAT medium. Why do the unfused myeloma cells die in this medium? Pause.
The unfused myeloma cells die because they lack the enzyme HGPRT, which is hypoxanthine guanine phosphorobabosyl transferase. The aminopterin in HGT medium blocks the main de novo DNA synthesis pathway. Therefore, cells must use the salvage pathway to survive. The myeloma cells lack the key enzyme for that salvage pathway, so they starve for nucleotides and die.
Perfect. Question 3. In a complement fixation test, the final result in the tube shows complete hemolysis of the sheep red blood cells, meaning the liquid is a clear pink color. Based on this result, is the patient positive or negative for the specific antibody being tested? Pause.
The patient is negative. Hemolysis means that free complement was available to destroy the indicator red blood cells. If complement was available at the end, it means it was not fixed or consumed by a patient antibody-antigen complex in the first step of the reaction. Therefore, the patient lacked the antibody, lysis negative.
You have to know that backwards and forwards. Okay, question 4. You are analyzing a flow cytometry dot plot to differentiate white blood cells. Which scatter parameter would be most useful for distinguishing a highly granular neutrophil from a non-granular lymphocyte? Pause.
That would be side scatter, or SSC. Forward scatter measures cell size, but side scatter measures internal complexity and granularity. Neutrophils are full of granules and have high side scatter, while lymphocytes are mostly nucleus and have very low side scatter.
All right. Question 5. A newborn infant is suspected of having a congenital toxoplasmosis infection. Which specific antibody class should you test for in the infant's own blood to confirm this diagnosis, and why is it the best choice? Pause.
You must test for IgM. The reason is that IgM is a large pentamer and cannot cross the placenta from mother to baby. Therefore, if IgM against toxoplasma is present in the newborn's blood, it must have been produced by the baby itself in response to an active in utero infection. IgG, in contrast, readily crosses the placenta, so its presence could simply reflect the mother's past exposure.
Last one, question 6. An initial ELISA screen for HIV comes back positive. Which serologic technique is considered the gold standard confirmatory test, which works by separating the viral proteins by molecular weight and identifying the patient's antibodies to each specific protein? Pause.
That would be the Western blot, also known as the immunoblot. Remember the mnemonic SNRW drop? Western detects protein. It provides specificity by confirming that the patient's antibodies react to a characteristic pattern of HIV proteins, like GP120, GP41, and P24.
That's it for today's deep dive into serologic diagnosis.
Keep reading those footprints and you'll find the beast.
We'll see you in this episode.